Good day, everyone, and welcome to another screencast in microbiology. I am your professor for today, Dr. Supachai Bassi. And for today, we will be discussing introduction to microbiology, and of course, we'll travel back in time and examine the historical background, okay, in the development of microbiology. Okay, so first of all, if you are a medical technologist and you will be actually specializing in the field of microbiology, then you are indeed a certified clinical microbiologist. And as a clinical microbiologist, you would play very important role in the clinical laboratory. First of all, you will be handling, culturing organisms from various clinical specimens. That's why you really have to take precautionary measure when it comes to biosafety and biosecurity. And then, of course, you'll be able to classify and identify microorganisms so that you'll be able to convey to the clinician the possible cause of the disease. And you'll also be involved in the prediction and interpretation of antimicrobial susceptibility testing. Because of your work in the laboratory, clinicians would be able to prescribe antibiotics properly. Hence, there would be improved treatment or improved management of the patient. So that's the role, basically the roles of medical technologists in a clinical laboratory. So let's talk about bacterial relationship to that of isolation. So when we say isolation, it means that from the clinical specimen, we'll be able to isolate the bacteria, which means that you really have to be familiar with the growth requirements of the microorganisms, such as the bacteria. Because by having this knowledge, this will allow microbiologists to select the correct medium for the primary cultivation of the microorganisms. If you're able to do such a thing, then the chances for the likelihood of you isolating the correct pathogen would be very high. So what are the steps in bacterial identification? Well, there are several steps. So first, you can also determine the morphology of the bacteria by doing the staining characteristics so that you'll be able to examine the cell wall structure, the size of the organisms, and even the shape of the organisms. And then there are also some procedures in the laboratory wherein you can actually observe the biochemical characteristics of these microorganisms. So at this particular juncture, we will now be discussing the overview of the microbial world. So first, let us, of course, travel back in time, okay, and take a look at the historical perspectives of the development of the science of microbiology. Now, this man is was Anthony van Leeuwenhoek. So, actually, he was not a microbiologist. He was a Dutch lens maker because at that time, microbiology is not yet known. It's not yet a known science. So, but he has some knowledge in biology. So, what he did, he discovered the so-called wee besties. So, because since he is a lens maker, he was able to invent this very crude type of microscope, and then he began to examine various pond water, hay infusion, peppercorn infusion, and then lo and behold, upon looking under his crude microscope at that time, he was able to see very small organisms, and some of them are actually motile. So at first, he actually called it animalcules. He called it animalcules. So at that time, the microscopic world was actually unknown. So what he did, he sent all his illustrations and drawing to the British Royal Society. So at that time, it was one of the most prestigious organization of scientists. And then of course, people at the scientists at the British Royal Society were actually amazed with his findings. So he became right away honorary member. So Anthony van Leeuwenhoek was then considered as the father of protozoology and bacteriology. Okay, so however, at that time,
Could you try again?
however, However, at that time, there was this ongoing debate over spontaneous generation theory. Scientists were divided into two sides: those who believe spontaneous generation theory and those who do not believe, or who did not believe over on the theory of spontaneous generation theory. So what is spontaneous generation theory? Actually, it's just a hypothesis, and according to them, living organisms can arise or could arise from non-living matter. So they call it spontaneous generation, and according to them, even without any empirical data to prove it, according to them, spontaneous generation has actually this element that can form life, this particular vital force. So those scientists who did not believe in spontaneous generation theory has this alternative hypothesis. Okay, they call it biogenesis theory, because according to them, living organisms could only arise from the pre-existing life. So you can just imagine the heated debates over whether spontaneous generation theory is correct or whether it is actually wrong. So in order to prove each point, they did experiment. So one of the earliest scientists who did experiment was Francesco Redi in 1666. So what Francesco Redi did, he filled 6 jars with decaying meat. So 3 jars were covered with fine net, and then 3 jars were left open. So since these were decaying meat, you'd expect that maggots would appear, okay, over those jars that were left open. So, well, of course, flies would actually come in touch over the surface of this decaying meat, and then of course, maggots were actually larvae of flies. So, however, when these bashers of Francesco read his experiment, they told him that, oh, you covered the jar. At that time, Antoine Lavoisier discovered the essential role of oxygen. Since you covered the jar, no oxygen actually come into contact with the decaying piece of meat, hence no maggots appeared. So Francesco Redi modified his experiment. So instead of totally sealing the jar, he just covered the jar with a gauze. So gauze will allow oxygen to come in and at the same time prevent flies from coming into contact with the decaying piece of meat. So what happened? Still no maggots appeared. But then those scientists who believed in spontaneous generation theory didn't stop. So one of their main proponents was John Needham. So imagine, the experiment of Francesco Redi happened in 1668. But in 1745, so after so many years and after so many decades, John Needham did an experiment. So what he did, he put boiled nutrient broth into covered flask. So you can see here in the illustration that there were two sets of this experiment. So the first flask were left open, and then the second flask were actually sealed. And then after several days, you could actually see growth, okay, over the first set of flasks, and there's no growth in the sealed flask. So what happened here? He said, according to him, microorganisms can still grow even on a sealed flask. Okay, however, upon examination of John Needham experiment, there was this one big flaw. And that flaw is that he didn't sterilize the glasswares. So since he didn't sterilize the glasswares, you would expect that contaminants would grow even if he actually sealed the broth. So this was actually proven by Lazzaro Spallanzani. So Lazzaro Spallanzani did the gravy is he had also 2 sets. So he actually used chicken broth. Both were boiled. Okay, after boiling on the same container, the first set, the flask was open. On the second set, the flask was sealed. So on the sealed flask, since he actually boiled the flask, okay, and used the same flask no organism actually occurred, as you can see in the illustration. But scientists who believed on spontaneous generation theory didn't stop. They said, since you use heat, the heat destroyed the so-called vital element of life, the vital force. So, in 1861, imagine, the debate started in 1668, but the debate actually ended when Louis Pasteur, okay, demonstrated that microorganisms are present in the air. So there were actually two sets, okay? So the nutrient broth were placed in the flask. It was heated but not sealed. Microbial growth occurs. And then the second set, the nutrient broth, okay, was placed in the flask, heated, and then sealed. No microbial growth. But this time, they didn't oppose Louis Pasteur. Perhaps John Needham was already dead by this time, but there was this very good empirical evidence why they didn't stop Louis Pasteur. And you know what? So Louis Pasteur did a unique experiment. So as you can see in the first set, he actually bended, okay, the curve of the flask, the tubing of the flask to be like a swan neck. So he called it a swan neck flask, wherein you will have a convoluted portion here. On the other hand, okay, Louis Pasteur, on his second set, cut this particular portion, the one that you can see here. So what happened? The curve of the flask prevents outside air from entering the flask. So no contamination occurs. While on the other hand, when the neck of the flask was broken off, bacteria reach the sterile broth and organism growth occurs. So until now, this particular swan neck flask is being displayed in Pasteur Institute in Paris, and Louis Pasteur becomes the father of microbiology. So beginning the discovery of Louis Pasteur, that's actually the start, the golden age of microbiology. So beginning with his work, discoveries included the relationship between microbes and disease, immunity, and even antimicrobial drugs. So Louis Pasteur actually contributed many things in the field of microbiology. First is fermentation. So Louis Pasteur was a French chemist. He wasn't even a microbiologist. So French are actually known to be one of the greatest wine connoisseurs in the world, probably because of Louis Pasteur. At that time, many winemakers were actually approaching Louis Pasteur, and they were asking Louis Pasteur for the solution, because whenever they are keeping storing their grapes for wine, because it will take years before you can actually produce a great wine, they were actually disappointed, because instead of getting a wine, they will actually produce vinegar. So what happened? Well, if you can remember the basic procedure in making wine, they will actually have to step on the grape itself in order to extract the juice. Now, the carbohydrates in the juice are supposed to be converted to a certain product. But if you'll be using the proper organisms. So there are several organisms that can participate in fermentation. But before that, let us define first, what do we mean by fermentation? So when we say fermentation is the anaerobic breakdown of polysaccharides, and there are two possible byproducts. One is alcohol, and the other one is acetic acid. Okay, it depends what is your starter organism. If your starter organism is a yeast, such as Saccharomyces cerevisiae. So at this point, I will be talking about scientific names. I will not be writing it. So guys, I want you to search for the correct spelling, okay? So the name of the yeast that participates or that serves as the starter organism in the process of fermentation in order for you to produce wine, beer. We call it Saccharomyces cerevisiae. It is the same baker's yeast that bakers are actually using. So if the winemakers are actually using Saccharomyces cerevisiae, after several years, they're supposed to expect alcohol. However, when the grape juice is contaminated by bacteria, okay, instead of having wine as the byproduct, they will have acetic acid instead. Can you just imagine the disappointment? They waited for 10 years only to find out that they produce vinegar. So, in order to improve their wine production, so Louis Pasteur invented pasteurization. And what is pasteurization? Pasteurization is the application of enough heat, okay, to destroy the bacteria, but at the same time preserve the grape juice and preserve milk. So just enough heat to kill the pathogen. And when these winemakers were able to pasteurize the grape juice and added a pure culture of Saccharomyces cerevisiae, after several years, they are now able to produce wine. And perhaps that is the reason why French wines are actually the best in the world. Okay? So, there you have it. Fermentation is the conversion of sugar to alcohol to make beer and wine. Okay? So, microbial growth is also responsible for spoilage of food. So, that's the reason why we do not want grape juice to be contaminated with bacteria, because instead of you producing wine, you'll be producing acetic acid instead. Okay, so Pasteur also demonstrated that this spoilage bacteria could be killed by heat that was hot enough to evaporate the alcohol in wine. So pasteurization, therefore, is the application of high heat for a short period of time. So in the later module, we will be discussing microbial control and the actual temperature and time requirement for the different methods of pasteurization. Okay, so because of the advances in the field of microbiology, many scientists began to discover many things. Let's say, for example, in 1835, Agostino Bassi showed that a silkworm disease was caused by a fungus. But 30 years later, in 1865, Louis Pasteur refuted it, and he said that another silkworm disease was actually caused by a protozoan. And then, in 1840s, Ignaz Semmelwise advocated hand washing to prevent transmission of puerperal fever from one OB patient to another. You can just imagine, at that time, doctors will be performing surgery or will be delivering a child without even washing their hands. But it was Ignaz Semmelwise who advocated hand washing. And the cases of post-puerperal fever and mortality rate related to giving birth were actually reduced. Okay. In 1860s, Joseph Lister used a chemical disinfectant to prevent surgical wound infection after reading at Pasteur work showing microbes are in the air. Microbes can spoil food and can cause animal diseases. So because he was able to read the work of Louis Pasteur, so he began to utilize chemical disinfectant during surgery. And that is the reason why Joseph Lister became the father of aseptic surgery. Okay. Now, there was another contemporary of Louis Pasteur. He was a German. So, his name was Robert Koch. Okay? Robert Koch proved that a bacterium causes anthrax. That's Bacillus anthracis. Okay? And provided experimental steps known as the Koch's postulate to prove that a specific microbe can cause a specific disease. Okay, so how do we know, or how do we place Koch's postulate in a model? So according to Robert Koch, okay, if the organism is healthy, okay, there's no organism in the blood, Okay, the presence of organism in the blood is called sepsis. So there's no sepsis. So it's actually absent. However, in a diseased organism, because at that time, Robert Koch was in the field and he saw a dead cattle, so the suspected organisms are found in the blood. So here, in number 1, the suspected causative agent must be absent from all healthy organisms, such as the one that you can see here, but it is present in all diseased organisms. So the diseased rat, if you're going to observe under the microscope, you'll be able to observe organisms are present. Number 2, the causative agent must be isolated from the diseased organisms and grown into pure culture. So you get the blood of the organism and you allow it to grow in the culture medium. Then you get another healthy animal. So the cultured agent must cause the same disease when inoculated into a healthy susceptible organism. So previously healthy organism was injected by the bacterial culture that came from the diseased organism. So what happened? The healthy organism died. And then you must be able to re-isolate the same causative agent from the inoculated diseased organism. This is Koch's postulate. So how are we going to apply Koch's postulate in our time? So when someone got the infection from a host, from other animal, you get sick, and then you can transmit the disease to a healthy host. This healthy host becomes sick, and then the organism from this healthy host can actually be injected into an animal, and this can also cause and this animal may also die from the original animal with the same disease. Okay? So this is somehow the clinical application of Koch's postulate. However, there are exemptions in Koch's postulate. Okay? There are some microorganisms that are unable to be cultured on artificial medium. Example, Treponema pallidum. Coronavirus, SARS-CoV-2. The coronavirus cannot be cultured. Therefore, they cannot be demonstrated using the Koch's postulate as a model. Then they could also be 2 or more organisms work in synergy to cause a disease. So sometimes when you have a disease, this could be caused not just by one organism, but other organisms. And then symptoms and diseases can be caused by any one of several microbes. So diarrhea can be caused not just by one microorganism, for example. So these are exceptions to Koch's postulate. Now let's talk about vaccination. So this guy here is Edward Jenner. Okay? So let me tell you first the story of Edward Jenner. At that time, smallpox is one of the most dreaded diseases. Okay? So nobody was actually spared from smallpox. It's one of the most dreaded diseases. So smallpox is caused by variola virus. Okay? Now, smallpox has a cousin. Okay? So the cousin of smallpox was actually a cowpox. And cowpox was actually the causative agent of cowpox was Vaccinia virus. So Edward Jenner, not Louis Pasteur, actually observed one of the milkmaids named Sarah Nilms. So Sarah Nilms was a milkmaid. So, and she was infected with cowpox. Okay? So, and Louis Pasteur observed that milkmaids with cowpox never get smallpox. So, why? Because, the thing here is, there is a principle of cross-reactivity. I'm sorry for the sound of the dog. Of the dogs, we have 2 dogs here in the house. So, the principle of cross-reactivity. So when you say cross-activity, if you get infected with cowpox, you produce antibodies. And these antibodies will also protect you against smallpox. So Edward Jenner needs to prove that this actually is correct. So there was this boy, James Phipps. Okay, so James Phipps is inoculated with cowpox pus coming from Saraneus. So Phillips fall ill with mild case of cowpox. So, kasi from the pus. And then, scabs are collected from smallpox patients. Imagine the risk of this particular examination, this particular trial. So, scabs were collected from the smallpox patient. So, Phipps is inoculated with the scabs of smallpox, but Phipps is unaffected and the protection is complete. You know what's the plot twist? So this is Edward Jenner, and then this is James Phipps. James Phipps was actually the son of Edward Jenner, so he experimented on his own son. So, however, because of that particular experiment, Edward Jenner was able to prove that if you will be inoculated with cowpox, you will be protected against smallpox. And Edward Jenner became the father of immunology. So vaccination is actually derived from the word vacca, which means cow. Okay, and then now we have the birth of the modern chemotherapy. So treatment with chemicals is called chemotherapy. So when you say chemotherapeutic agents, these are actually being used to infectious disease, and these chemotherapeutic agents can either be synthetic drugs or antibiotics. So when you say antibiotics, these are chemicals produced by bacteria or fungi that inhibit or kill another microorganisms. So the term antibiotics literally means against life. So the first chemotherapeutic agents were actually discovered by Native Americans. Okay, so they actually called it the quinine tree from a cinchona tree. So this quinine from the cinchona tree bark was long used to treat malaria. But the first chemotherapeutic agent, synthetic chemotherapeutic agent, was discovered by Paul Ehrlich. Okay, a synthetic arsenic drug called salvarsan to treat syphilis. Do you know that salvarsan was also known as 606? Why 606? Because this is the compound number 606, because the 605 other compounds didn't work. So on his 606th trial, imagine the perseverance of Paul Ehrlich. He was able to successfully formulate salvarsan. And why do you think it's very difficult to formulate salvarsan? Because of the principle of selective toxicity. You do not want the compound to be harmful to the pathogen and at the same time harmful to the host as well. You just want it to be harmful to the pathogen, not with the host. And in 1930s, sulfonamides were also synthesized. However, it was only in 1928 that the first naturally occurring antibiotic, was discovered by Alexander Fleming. So Sir Alexander Fleming, because he was knighted after this discovery, was actually accumulating petri dishes in the lab. And then his culture, which is a Staphylococcus aureus, got contaminated with Penicillium notatum. So he found out that the colony of Penicillium notatum inhibited the growth of Staphylococcus aureus. Because of this, he got that idea that this penicillium can inhibit the growth of Staphylococcus aureus because of the presence of the zone of inhibition. And he extracted the active ingredient of Penicillium notatum and named it penicillin. So in 1940s, penicillin was tested and clinically and was actually mass produced in time for the Second World War. Okay, now because of the modern development in microbiology, so we have now branched out into different field of specialization, such as the course that we have right now is bacteriology, and then there's another course called mycology, or the study of fungi, and then parasitology, or the study of protozoan and parasitic worms, and then there had also been advances in genomics, the study of an organism genes, which have provided new tools for classifying microorganisms. So immunology also became one of the most important science here. So immunology is the study of immunity. So there you could actually see development of vaccines and interferons. So these are being investigated to prevent and cure viral diseases. So the use of immunology to identify some bacteria according to serotypes was originally proposed by Rebecca Lancefield in 1933. And that is the reason why if you will be studying Streptococcus, you will have Group A Streptococcus, Group B Streptococcus, Group C Streptococcus, Group D Streptococcus. All of these are actually considered as the Rebecca Lancefield classification. Okay, so also in microbiology, the study of viruses have actually improved. So viruses, the recombinant DNA technology is also improved. So when you say recombinant DNA, it is the DNA is actually made from two different sources. In 1960s, Paul Berg inserted animal DNA into the bacterial DNA. So that bacterial DNA is called the vector. And the bacteria can now produce animal protein. So we can have recombinant genetically engineered insulin, genetically engineered factor VIII. So recombinant DNA technology or genetic engineering involves microbial genetics and molecular biology. And there have been many developments. So the 20th century is considered to be as the golden age of microbiology. So you can see here, George Biddle and Edward Tatum showed that genes can encode a cell's enzymes. Okay, Oswald Avery, Colin MacLeod, McLean McCarthy showed that DNA was the hereditary material. Francois Jacob and Jacques Monod discovered the role of mRNA in protein synthesis. One of them is actually puffing cigarette in the laboratory. Do not do that, okay? And these are the selected Nobel Prizes in physiology or medicine involving microbiology. So as you can see, Robert Koch here discovered Mycobacterium tuberculosis. Elie Metchnikoff, okay, discovered the role of WBC in phagocytosis as early as 1908. Alexander Fleming won Nobel Prize for penicillin. And then Prusiner was able to discover prions. Okay, what are prions? So I will be including prions in the animations on prions in the future modules. So take a look. What are prions? Prions are even smaller than viruses. So that's the end of introduction in microbiology, okay, which talks about history or historical perspectives. Again, this has been Dr. Rebecca Bassi saying, always stay safe and may God's blessing be upon each one of you. Bye!